Before talking about meiosis in more detail, we need to touch on the differences between mitosis and meiosis. Mitosis is a single nuclear and cellular division. Meiosis has two divisions. After mitosis, the chromosome number in the newly formed cell is the same as the original cell. After meiosis, the chromosome number in newly formed cells is reduced by half. Mitosis produces genetically identical cells. Meiosis produces genetically different cells.
Prophase 1 is divided into 5 different substages. The 5 different substages are leptotene, zygotene, pachytene, diplotene, and diakinesis. In the leptotene stage, chromosomes condense and become visible. In zygotene, homologous chromosomes pair up and begin what is known as synapsis, or a very close pairing. Oftentimes during this, you may hear the words tetrad or bivalent. And when that is said, that just means a homologous pair of synapse chromosomes consists of 4 chromatids. During pachytene, the chromosomes will become shorter and thicker. In diplotene, the paired chromosomes begin to separate into 2 pairs of chromatids. And finally, to cap off prophase 1. During the last stage, called diakinesis, the separation of homologous chromosomes is complete and crossing over has occurred. We see crossing over take place in prophase 1, and that is basically just the homologous chromosomes exchanging genetic information. Prophase 1 sets the stage for metaphase 1, with the nuclear membrane breaking down and the spindles forming.
Metaphase 1 is initiated when homologous pairs of chromosomes align on the metaphase plate using independent assortment. Anaphase 1 is marked by the separation of homologous chromosomes. The 2 chromosomes of a homologous pair are pulled to opposite poles.
In telophase 1, the chromosomes arrive at spindle poles and now can begin to unravel to the relaxed chromatin state as the nuclear membrane begins to reform and microtubules dissolve. This is followed by cytokinesis, where the cytoplasm divides, forming 2 genetically different haploid cells. After this is interkinesis, a period of time between meiosis 1 and meiosis 2, but is different from interphase because no DNA replication takes place. After this rest period, the 2 cells enter meiosis 2. The first step of meiosis 2 is prophase 2. Similar to prophase 1, the nuclear envelope dissolves once again and the genetically different chromosomes condense. The centrosomes also duplicate and begin to migrate towards opposite ends of the cells. Next is metaphase 2. The dense individual chromosomes align on the equatorial plate. As in metaphase 1, independent assortment occurs, and the chromosomes line up randomly and independent of each other, adding another level of variation, and microtubules attach to the chromosomes at the kinetochores.
In anaphase 2, the kinetochores of the sister chromatids separate and the chromatids are pulled towards the opposite poles. Each chromatid is now a distinct chromosome. In telophase 2, the chromosomes arrive at the spindle poles, the spindle breaks down, and the nuclear envelope reforms around the chromosomes. After the final phase of meiosis 2, the cell goes through cytokinesis where the cell divides. By the end of meiosis, the products that are left are 4 haploid gametes that have their own unique set of genetic material that originated from the original cell.